Yeah. Okay, fine. Okay. So time is 9 o'clock. We will start sharp. Okay. So what we are going to do is I'm going to just show you all the questions of last 6 papers. Okay. That is, we'll start from May 2022 and 22 May and November paper. And then 23 May and November paper, 24 November and May, and 25 the last exam, May 2025. Okay. So the overall, whatever questions have been asked in the last 7 papers, we will have an idea. Okay. We'll not go into much details. We have a lot of questions to cover. Wherever explanation is required, I will be teaching you that. Okay. So fine. So first question, DNA protein interaction is studied using, the answer is DNA footprinting. Okay. So basically DNA fingerprinting is a technique to find out RFLPs. ELISA is a technique, you know, it's an immunoassay, basically for antigen antibody reactions. And northern blot is a technique for RNA hybridization to identify that. Okay, so the DNA and protein interaction is studied. Answer is DNA footprinting. Okay, so we are starting with May 2022. Okay, so a patient with Ewing sarcoma presented with EWSR1 gene mutation and technique shown in the image was performed. Upon fusion, yellow fluorescence was seen. Identify this technique. Okay, so this technique is nothing but the fluorescence in situ hybridization. Yes, correct answer. See, you can see the word fluorescence. They have already given you, it is EWSR1 gene mutation. So it is a known mutation. So I already told you only known mutations, amplifications, micro deletions, or translocations. These can be detected by fluorescent in situ hybridization. This is immunohistochemistry, which is wrong. This is RT-PCR is also wrong. And Sanger's is DNA sequencing. This is also wrong. Okay. So this is the answer is the fish. Okay, fine. Which of the following is correct regarding the enzyme inhibition shown in the picture? Now it is very, very easy. I have already taught you this. When you see there is a change in the x-axis, see, with inhibitor and without inhibitor, there is a change in the x-axis. There is no change in the y-axis. That means that there is no change in the velocity. So what has changed is here Km has increased. Now you should not think that sir told Km has increased, but with inhibitor it is coming this side. This is the inverse plot, double reciprocal plot, 1 by plot. So as we are going this side, it is increasing. That is the first thing. And regarding the Vmax, there is no change. So regarding the Vmax, there is no change. So the answer will be competitive inhibition. The remaining options are not fitting into the picture. Okay. In a patient with maple syrup urine disorder, all the following amino acids should be restricted in diet except. So maple syrup urine disorder is a branched chain amino acid disorder where there is a alpha keto acid dehydrogenase defect or deficiency. That means there is a accumulation of branch chain amino acids. So what you need to do is branch chain amino acid restricted diet. That means valine, leucine and isoleucine are wrong. The only thing which can be given is methionine. Fine. Very nice. Okay. Next one. Deamination of methyl cytosine forms. So this also have shown you in the class. So in our workbook also we have shown you. Let me open the workbook. Just to give me a minute. So this is the page. So this is the page. Okay. What is the question they have asked? Deaminated methyl cytosine. Okay. Deaminated cytosine. Deaminated. Amino group is removed. So deaminated cytosine is called as uracil. When you are putting the methyl group over there, then it will be called as a thymine. I have taught you this point already. So what is the answer here? Deamination of methyl cytosine forms thymine. Thymine will be the answer. Okay, fine. All are true about Human Genome Project except it took nearly 2 decades. Yes, 1998 to almost 2013. So nearly 2 decades. Yes. And then next generation sequencing was used. That is also correct. They came to next generation sequencing. It took international US Department of Health and this thing. The only thing is the total number of genes that was found was 19,900. It is not 30,000. Okay. So this will be the correct answer for this question. Again, this question, some of the options, some of the students are telling differently, but this is what finally I got. So among these 4 options, if you want to go to the best option, this is 100% wrong. Total genes is 19,900, should be 20,000. So probably that will be the correct answer. And then banding technique, which is used for dicentric chromosome. Dicentric chromosome is C banding. Dicentric chromosome means the chromosome which has 2 centromeres. Okay, so it has 2 centromeres. So it is visualized by C banding. G is Giemsa banding and R is reverse banding. This is more random. Q banding is the another technique. Okay, quinacrine banding. That will be the third technique. Okay, so the answer here is C banding is the correct answer to visualize dicentric chromosome. Right. In Sanger sequencing, di-deoxynucleotides are used because, okay, so I told you we have a 5-carbon ribose. If you remove a OH from the second carbon, it is called as a deoxy. The same, if you remove from the third also, then it is called as dideoxy. When you add the deoxy, it will elongate or it will further the replication. When you add the dideoxy, it will terminate or stop. Sanger used this technique of di-deoxynucleotides. Yes, very good. All of you are giving the correct answer. Because di-deoxynucleotide binds and causes breaks so that DNA polymerase cannot act further. So it terminates polymerization. Can anyone of you tell you what is the other method? Other than Sanger sequencing, there is one other scientist who also came up with a method for DNA sequencing. Anyone? Can you put it in the comment box? It is the Gilbert chemical sequencing method. Okay. So Gilbert was another scientist, okay, who came up with the, yeah, correct answer. Very nice. Fine. Now, misincorporation of uracil into DNA is seen in deficiency of. So, what exactly is this question is? It is asking you about the conversion of DUMP to DTMP. So, this reaction is not happening. So, uracil is entering into the DNA. So, what could be the deficient factor? So, for this reaction to occur, you need to add a methyl group. Who is the methyl or 1-carbon donor? The 1-carbon donor is folate. So this dihydrofolate has to be converted into tetrahydrofolate. Tetrahydrofolate has to get 1 methyl group from the serine to glycine reaction. And this is given as the methyl group. So what is happening is when there is no folate, this reaction is not happening. So all the uracil is going to sit in the DNA, which is defective, right? So the answer here would be folate. So misincorporation of uracil into DNA is seen in the deficiency of folate. So which is incorrectly paired, very, very high yield question. Okay, straight away, you know, lipase pancreatitis is correctly paired. This is called as the LDH flip phenomenon, which I have taught you. Ceruloplasmin hepatocellular generation is a Wilson's disease. So that straight away rules out beta-glucocerebrosidase. Beta-glucocerebrosidase is the Gaucher's disease. So here it is given the von Gerkes. So this will be the answer for this question. This is the incorrectly paired. Which of the following is not used for protein precipitation? See, protein precipitation techniques, many techniques I have told you. Predominantly, proteins are precipitated at isoelectric pH. Other than that, proteins are precipitated during heating. You know that, right? Milk heat coagulation test. Okay. And then while adding the strong acids or alcohol. And then there is acetone, which is a solvent that is also precipitates the protein. Finally, we have the heavy metals. So these are all protein precipitators. These are all protein precipitators. Okay. So what is this trichloroacetic acid? This is called as an alkaloidal reagent. So this also can be added along with something called as a sulfosalicylic acid. Sulfosalicylic acid. So sulfosalicylic acid also can precipitate the proteins. So here what they are asking you is which one is the answer? The answer is this one. Change in the pH other than isoelectric pH. No, isoelectric pH only causes the protein precipitation. At that pH, it exists as a Zwitter ions where the net charge on the molecule is 0. Okay, fine. G6PD deficiency can lead to all except decreased membrane lipid peroxidation, decreased fatty acid synthesis, decreased NADPH generation or glutathione, scavenging RBC by macrophages. Now, what is the reaction of G6PD? Glucose 6-phosphate is converted to gluconolactone. Now, in this reaction, NADPH is produced. This NADPH powers the glutathione. Glutathione reductase, yes. Glutathione is converted into glutathione, reduced glutathione or active form. This is oxidized. Now, this will power the enzyme which is called as a glutathione peroxidase. So this will be active. So this will become active. Now, once it becomes active, this will destroy the peroxides. So protects the RBCs. Protects the RBCs. So what happens here is they are asking you G6PD deficiency causes all except. So when there is G6PD deficiency, there is no NADPH, there is no glutathione active, there is no glutathione peroxidase working, which means that H2O2 will increase. This is called as a peroxide. So when G6PD is deficient, peroxides will increase. That means this option is totally wrong. Decreased membrane lipid peroxidation. It is actually increased peroxidation. So this will be the answer. What about the remaining answers? See here, decreased fatty acid synthesis. Yes, because for fatty acid synthesis, you need NADPH. No NADPH, no fatty acid synthesis. Decreased NADPH, straight away they gave you. Scavenging RBC by macrophages cannot be done. That is because we cannot, peroxides are keeping on increasing. Okay, so that scavenging action is also gone. So answer will be decreased lipid peroxidation is wrong. It is increased lipid peroxidation. So May 2022, INICET had 11 questions from, no 11, 12 questions from biochemistry. All the 12 are done for us. Okay, so that's over. Now let's move on to the November 2022 paper. Straight away, what is the diagnosis? Now, what is this? This is the orange tonsils. Where do you see orange tonsils? When there is a ATP binding cassette gene 1 mutation leading to a defective transporter. So that will cause decreased HDL in circulation. So what is this disease called? This disease is called Tangiers disease. This disease is called Tangiers disease. So the answer here for orange tonsils is Tangiers disease. Now, glycogen phosphorylase is activated directly or indirectly. Glycogen phosphorylase is the rate limiting enzyme for glycogenolysis. What are all the MCQs about this? This enzyme has a cofactor which is vitamin B6. That's the first MCQ, JIPMER MCQ. The second one, who increase and who decrease the activity of this enzyme? Who increase this activity? It is calcium and ADP. Who all decrease the activity of this enzyme? In the muscles, it is ATP. In the liver alone, it is glucose. And then there is glucose 6-phosphate, which is common. Okay, so that's all. This is all that you need to remember. And of course, hormonal wise, always insulin decreases the glycogenolysis and glucagon increases. So now you just see glycogen phosphorylase is activated by who? Very simple answer. Among these three, one person. Who is that person? It is the calcium. Okay, remaining all of them will go for inactivation of glycogen phosphorylase, right? So burnt sugar smell is because of defect in, very simple, burnt sugar smell is a burnt sugar smell is because of MSUD. Okay, so burnt sugar smell, MSUD. What is the defect in MSUD? It's a very straightforward question. Nothing is there. Branch chain keto acid dehydrogenase. There's one other point. This vitamin B1 dependent. There are other two enzymes, alpha KGDH and PDH. These are all vitamin B1 dependent enzymes, right? Cystic fibrosis is caused by which deletion? It is delta F 508, where F stands for phenylalanine. So the answer is straightforward. Phenylalanine at 508 is a deletion mutation which causes cystic fibrosis. Cystic fibrosis is a chloride channel defect. Right? Fine. Significance of HMP shunt are all except. So HMP shunt is very crucial because it acts as a flux pathway for glycolysis. 2, it protects the RBCs. 3, it generates special product NADPH. 4, it generates ribose 5-phosphate. 5, 0 ATP produced or used. These are the 5 points you need to remember. So it is a source of NADPH. It is a source of ribose 5-phosphate. Of course, it utilizes ribose to generate the ribose 5-phosphate. So what is the answer? It is not a source of acetyl-CoA. So where does acetyl-CoA comes from? Acetyl-CoA comes from glycolysis. Glycolysis end product is pyruvate. Pyruvate is converted to pyruvate dehydrogenase complex to acetyl-CoA. It is not coming from HMP shunt. Very simple and straightforward question. Refsum's disease is caused by defect of. So what is Refsum's disease is caused by? It is the defective conversion of phytanic acid to pristanic acid. Now this is a very long chain fatty acid. So the enzyme name is phytanyl-CoA hydroxylase. So because of this, some phytanic acid accumulates in the brain, it accumulates in the eyes, and then there is also anosmia, there is also partial blindness, deafness, neuropathy, paresthesias, all those things. Now all this is happening where? It is happening in the peroxisomes. In the peroxisomes, the VLCFA is a special type of oxidation. It is called as the alpha oxidation. So alpha oxidation of branch chain. Sorry. Yeah. Alpha oxidation of branched chain fatty acids, in the sense it is the other name for VLCFA. Okay. That's all. Right? Good. So SARS-CoV-2 diagnosis is done by all except. So RT-PCR is there, real-time PCR is there, immunoassays is also there, southern blotting is not there. Southern blotting is nothing but it is not for diagnosis, it is for DNA hybridization technique. It's a very simple question. So southern blotting will be the answer for this. Okay. Now, selenocysteine is present in which of the following? Selenocysteine or Basically, they are asking you, selenium is a cofactor for three enzymes: glutathione peroxidase, deiodinase, thioredoxin reductase. So which is the answer here? Thioreductase peroxidase wrong. Thioreductase oxidase wrong. Glutathione reductase is also wrong. Okay. For glutathione reductase, it is vitamin B2, which is the marker. So glutathione reductase deficiency marker is vitamin B2. Here, selenocysteine, answer is glutathione peroxidase. Okay, fine. Now, in the November 2022 paper, the most difficult question was sirtuins. Now, sirtuins are basically, this was like out of the box, totally nobody had any clue about that. Okay, so sirtuins is nothing but they are actually histone modifiers. What they do is they cause histone deacetylation. You all know that when there is histone acetylation, it actually increases euchromatin. Now, histone deacetylation increases heterochromatin. Histone deacetylation increases heterochromatin. Is that okay? Fine. Give me a minute. So here, that is one thing. So it is histone deacetylase and causes chromatin condensation. This is correct. Number two is this histone modifier sirtuin increases glucose control. How it does means it potentiates insulin. It potentiates insulin and increases glucose control, and it also regulates stress and aging. So that means that it increases the lifespan. So this is also correct, and this is also correct. Now that leaves us with the last option. It is stimulated by niacinamide, even though we don't know anything about that. But then sirtuins are increased by NAD and it is decreased by the product of NAD and NADP, which is the niacinamide. So actually, it is not stimulated by niacinamide. It is actually inhibited by niacinamide. So many of you have put the answer correctly. Very good. Very good. So keep it up. Fine. Now, choose the next right sequence. This is exactly what I taught you in the class. What is the correct sequence? I will show you the picture. So you all know the pathway, right? So this is our pathway. Okay, fine. So what is the first enzyme? The first enzyme will be desmolase. That's what I taught you. Side chain cleavage enzyme. The other name is desmolase. Okay. And then we have the 3-beta hydroxysteroid. In this question, it has to go in this direction or it has to go in this direction. We are going in this direction. Okay. So from desmolase, it's 3-beta HSD. Then it is 17-alpha hydroxylase. Then it is 17-20-lyase. This is what has been asked in this particular question. So 20-22 desmolase, 3-beta, 17-alpha, 17-20-lyase. Remaining everything is wrong. See, first 3-beta and then lastly desmolase, wrong. Desmolase, but after that lyase is coming, wrong. 17-alpha is beginning, lastly desmolase, again wrong. So that leaves us with only one option, which is the first option. Okay, is that clear? Fine. 2,3-BPG causes unloading of oxygen to tissues because it binds to the central, it does not have any link to ATP production. Okay. So it binds to the central cavity of hemoglobin surrounded by histidine and lysine. You all know that which histidine is involved in oxygen binding. Can you tell me? It is called as histidine E7. Okay, so it binds to the central cavity of HB surrounded by histidine and lysine and promotes TOT structure. What is TOT structure? It is the deoxygenated form. So this will be the role of 2,3-BPG. The other point is, where is 2,3-BPG formed from? It is formed from Rappaport-Newbery cycle. Is that clear? Fine. Now next question. Where identify the left curve. So we have got this curve is called as the ODC curve, oxygen dissociation curve. Okay, there is one left curve, there is one right curve. The left curve is a parabola. The right curve is a sigmoidal curve. So they are asking you what is the left curve. So you all know that the left curve is nothing but the myoglobin. And you can also see from the picture, it is, this thing is 26 and for this it is will be 8 mmHg. So that is called as the P50. Okay, fine. So the P50 of myoglobin is lesser than the P50 of hemoglobin, which means that myoglobin has more affinity to oxygen than hemoglobin, right? Fine. Assertion. This type of questions is not coming nowadays, but before they were asking. One statement will be there, one reason will be there, then you have to find out which is correct, which is wrong. Assertion, PKU has a mousy order. Phenylketonuria has mousy order, which is correct. Tyrosine and norepinephrine are not formed in PKU. Tyrosine comes from phenylalanine. So phenylalanine gives rise to tyrosine. And norepinephrine comes from norepinephrine, not only norepinephrine, dopamine, norepinephrine, and epinephrine. All of them come from tyrosine. So in this step, there is a problem. That means that there is no tyrosine, there is no catecholamines. So that is also a correct. So both A and R are true. But is R the explanation for A? No. PKU has mousy order because of the accumulation of phenylpyruvate, phenylacetate in the urine. That is the reason that is coming from the phenylalanine, accumulation of phenylalanine. So it is not a problem because of the lack of tyrosine. It is a problem because of excess phenylalanine. So both A and R are true, but R is not the correct explanation of A. That is the answer. Okay. So next, Zellweger syndrome is caused by defect in Zellweger syndrome. Straight away, it is a peroxisomal biogenesis disorder. So there is nothing to think there. Zellweger syndrome is caused by defect in the peroxisomes, right? Long-term glycemic control in a person with hemolysis is assessed by fructosamine. First of all, long-term glycemic control for a period of 3 months is assessed by HbA1c. But in this particular case, it is hemolysis. So hemolysis, HbA1c values are not stable. So we can't rely on them. So they are using another parameter which is called as fructosamine. What is fructosamine? Now fructosamine is a protein. Fructosamine is a protein. Just like how HbA1c is a protein, fructosamine is also a protein. Okay, fine. Is that okay? It is actually a glycoprotein. It is a glycoprotein. Fine. Right. Which of the fructosamine can detect for 1.5 to 2 weeks glycemic control? So HbA1c is for 3 months. This is like 1.5 to 1.5 to 2 months. Six weeks you can keep like this. Don't wait. 6 weeks. Just keep it like this. 6 weeks control, glycemic control can be monitored by fructosomy, right? Okay. So which of the following tissues are insulin dependent? Very simple. They are just asking you what are all the GLUT4 dependent. There are only 3 fellows who are GLUT4 dependent. 1 is skeletal muscle, 2 is cardiac muscle, 3 is the adipose tissue. So skeletal muscle, cardiac muscle, and the adipose tissue. All right. Pooch tragedy. Pooch tragedy is a tragedy which is happening because of illicit liquor. Probably this is a community medicine question, or this is a ARAC or forensic medicine question.